Okay, welcome back. Today we're gonna talk about the calcium regulation of contraction. These are the objectives of Fang's second lecture, and what we need to understand is how calcium regulates this contraction, how calcium will react and interact with tropomine to also create this contraction, and then what troponin and tropomyosin even are. So those first three objectives are all on this side, and then the last two we'll cover later. So for starters, troponin is a component of this tropomyosin. It's going to control what it does. It's basically the boss of tropomyosin. Tropomyosin is just there, just reacting, just living, and then troponin will tell it when to move, when to come back, etc. So when we are relaxed and the calcium in our muscle cells are absent, this tropomyosin is going to be blocking the binding site for this cross bridge on this myosin. Remember, this is myosin filament and this is actin. And we're talking about when myosin is going to pull actin onto itself. So when tropomyosin is blocking that active site, this cross bridge can't happen. It can't bind and it's not going to contract your muscle. So our muscles are relaxed. When we do want to contract our muscle, that's when calcium is going to come into play, and calcium is going to bind to this troponin, and it's going to do that calcium troponin complex. That is going to move tropomyosin out of the way, and it's going to expose that binding site. So now that cross bridge can happen, and we can attach, and we can do the power stroke, and we can do everything that I explained in the past video. So this is basically just saying what I just said. So those were the first three objectives, just right there. So now the last two objectives are understanding what SR is, and then understanding the relationship of the triad. So SR is the sarcoplasmic reticulum, so it's the smooth ER endoplasmic reticulum, the smooth endoplasmic reticulum of muscle cells, sarcoplasmic reticulum. So that is going to store the calcium needed to create this. So once this is done, calcium is going to go back to the sarcoplasmic reticulum. Once it's needed, it'll come from that storage back over here. And then sarcoplasmic reticulum is kind of like a covering around that muscle cell. And then that covering is going to have a thicker extension in that A band and I band junction. Is going to have a thicker extension, and that's called the terminal cisternae. And that terminal cisternae, there's going to be two terminal cisternae that are going to connect in this T-tubule in the middle. That T-tubule is actually part of your muscle cell. It's that plasma membrane of the muscle cell that just kind of creates an invagination or a tubule of that plasma cell. So the two terminal cisternae and the T-tubule are going to create the triad. So what the triad itself does, all these things together are going to facilitate action potential coupling, which I will explain in the next video. But that's what the triad is going to do. It's going to facilitate the coupling of the action potential. But the T-tubule, and specifically, remember I was saying that it's the membrane coming into the cell, is going to allow that action potential to travel deep into the cell itself. And then the fun fact for this topic is that the generation of this cross bridge itself, these little myosin heads, will need both ATP and calcium. ATP is needed for both the relaxation and the contraction of muscle cells, but then calcium is only needed for the contraction itself. And that's all I have.